are some of the things that we're going to be seeing in this chapter. We're going to be talking about mutations, um, and some other things that come up when we have, obviously we're going to be talking about Hardy-Weinberg, like I was telling you, how to measure these changes in populations, but also, what causes natural selection, or, if we think about mutation as the mutation produces a lot of variation, and natural selection is the one that edits the variation and sorts it out for us. So, looking about how these mutations can occur, but also what, genetic drift happens, which is where we see populations where neutral traits start to wander. And it kind of, it's measurable and we can see it. And we'll talk about some things like the bottleneck effect, which may sound familiar to you, the founder effect, which might sound familiar, and some things like that. So first, this picture is in your textbook and it's showing a snail species, Cypea normalis, which you see here. It's native to Scotland. They're beautiful, kind of like a nautilus. But they're different. Their variation in the shell color here may have adaptive value in these snails, because some colors predominate in cooler environments, whereas other colors are more common in warmer environments. So they're not 100% sure yet. Again, this study is always, science in general is always changing, right? But evolutionary science, every little new thing we find, new organism, new fossil we find, is adding to this big picture. So evolution occurs in populations, not individual. It's not an individual thing. Obviously, a person can evolve, and by their outlook on life, their job, their all of that, their family situation, that can evolve. But as a species, evolution happens as a population changes, and I think that that's important to remember. If you guys remember talking about how humans are not the end goal of evolution. We're just a mere little, piece of the puzzle here. All populations go through evolution. So individuals of the same species living in a particular place at the same time is what we consider a population, remember. And they vary in characters. They vary in characters, meaning like what you had to plot on the phylogenetic tree or the cladogram you guys did on your exam. So those traits vary, okay? And biologists study variation in a particular character by taking measurements of that character in a population. And they try to do this quantitatively as much as possible, as opposed to qualitatively. However, quantitative does give you some room for error. Okay, so, quantitative is always valued more in science. But in some examples, especially when you're studying certain characteristics, you can have a little bit of issues with that, because, like intelligence, measuring intelligence. I think we all can appreciate that intelligence comes in many, many forms. And there have been so many different tools that we've created to try and measure intelligence. Obviously, IQ testing is is a predominant form of intelligence testing. But we know that people, for example, we talked about shows that we like to watch. I like shows where I can see people being artistic, because I have like zero art intelligence when it comes to painting, drawing, and things like that. I can't make sense of that in my brain, whereas an artist can't make sense of some other things in their brain, but they understand art like I understand science. All right, so intelligence is one of those qualitative characteristics that is very hard to quantify. And biologists, intelligence is a part of evolution that we do try and measure. Okay, we're always trying to see how intelligent species are, and compare that to, ancestors and things like that, like human intelligence as opposed to Cro-Magnon intelligence, as opposed to Neanderthal intelligence, and things like that. Okay, so a little bit of history here with population genetics, which is really what this chapter is talking about. It's evolutionary change in population. So population genetics, Darwin, did not really get to touch on much population genetics because we didn't understand genetics. So this is like a post-Darwinian time that this has come about. And in fact, Darwin, remember in our history of evolutionary ideas here, Darwin actually had an unread copy of Mendel's papers on his desk. He had a copy, but no leading scientists appreciated Mendel in his lifetime. And it wasn't until a bunch of botanists actually independently rediscovered Mendel's work in 1900 in that time, and started a new discipline, which we now call genetics. So Darwin, talking about evolution, obviously didn't have that genetics piece. And now there's this whole field of genetics called population genetics. All right, population genetics is the study of genetic variability within a population and of the evolutionary forces that act on it. All right, so it is possible to estimate the amount of observed variation that is genetic as represented by the number and frequency and kinds of alleles in a population. We can actually look at a population, look at a certain trait, look at all of the phenotypes that exist for that trait, and calculate and quantify these things and predict what will happen. And think about the implications of studying genetics of a population. Understanding diseases, understanding medicine, things like that. Obviously, yes, those are huge for understanding this. Like, for example, understanding how, if there's a link between type 2 diabetes and is there a genetic link to it? Are you genetically predisposed? And if more and more people in America are getting type 2 diabetes, our population is shifting somehow. All right. And studying that. So obviously, epidemiology, pathology, these are areas where population geneticists are going to be necessary. But think about in terms of conservation of the environment, okay? Studying how habitat fragmentation or habitat destruction, deforestation is a huge thing, things like that, how that's affecting the genetics of a population. Because if we have mass, it's almost like we're forcing these extinctions, right? And we're forcing animals to now be endangered. And when we do that, we're creating a bottleneck effect, which we're reducing the genetic variation amongst the population. We need to be able to realize what that will do to that population. And that's one of the things that a population geneticist would do. So some traits, it's important to remember, some traits don't help or hurt reproduction. Sorry, my dogs are trying to get in here. So some traits are neutral. And we can actually study those traits and track those traits in a population. So, for example, if I'm trying to study how a population, sorry, the dogs were just trying to get in. So. If I'm trying to study how a population will be affected by a trait, and that trait makes that animal more likely to survive, okay, like a thicker coat or something like that. I'm looking out at this snowy weather, and we've seen some deer that are like super fluffy right around, the other day. We saw some yesterday or Saturday that were super fluffy deer. Let's say a really thick, fluffy coat, is something that we're trying to trace, and we're trying to study a deer population. However, that trait is tied to the reproductive success of that, or not the reproductive success, the survival success of that deer population. So we might not get a true picture on how natural selection is working here if the trait that we're studying is always linked to reproductive success. It's almost like we need another kind of trait to study that doesn't affect anything and that is more of a neutral thing. All right, so we can track those types of genetics, those types of traits, to study how a population would normally evolve. It's, without that trait being an indication of success reproductively or survival. So fixation of neutral genes. Fixation refers to like 100% frequency. All of a sudden that gene is now everybody we see has this gene. Um, we can and fixation of a neutral gene is almost like radioactive decay in how we use it to study. So like radioactive decay, we know when we find a fossil, we're going to study it, and we know that we're going to use some sort of like carbon 14 or whatever, an isotope, radioactive isotope, and we're going to see how long this thing has been in existence based on the amount that it has left in its specimen. We can almost track the evolution of a population by tracking the fixation of a neutral gene. Because that gene doesn't affect the livelihood of the animal, it is a neutral gene. And so with those neutral genes, you might say, why would they evolve anyway? What is a neutral gene? And think about it like some DNA we know does not affect phenotypes, right? They're not expressed. Some DNA is not expressed. So gene duplications over history have created something called pseudogenes, right? This is a concept not in your textbook because again, this is like a little bit more of a complicated thing, but pseudogenes they're not expressed, but we contain them in our DNA. And gene duplications are, if you remember, duplication is a type of chromosomal mutation. So where you take a section of a chromosome and there's a duplication of it, and all of a sudden you have a whole other set of the same nucleotides. So in these genes that aren't expressed, we just can sometimes get duplications and duplications and duplications, and now we have a bunch of these genes that are not expressed. All right. Some other mutations don't affect amino acid sequence, and we call that wobble. So wobble is why, if we look at, let me pull up a genetic code here. We might not have learned the name for wobble, but wobble is why we see, let's see here, why we see that UUA codes for leucine and UUG codes for leucine. Okay, that's called wobble. All right. And that's the reason why we see, a lot more flexibility with that third letter of a codon because of this concept called wobble. All right, that means that some mutations just aren't going to cause a change in amino acid sequence. The amino acid sequence, even if the DNA gets mutated, could be fine. Some amino acids, let's say the genetic code gets mutated, the DNA gets mutated, calls for a different amino acid sequence, okay, and that different amino acid sequence has an amino acid in it that is it's actually very similar to the amino acid that was supposed to be there. So amino acids, if we look here, classifying amino acids, all right, you're going to see that we can categorize them into groups. All right, so I'm actually just gonna delete this here. All right, so this is wobble there we're talking about, and here is our amino acids. Okay, we can put them into groups based on their charge. Okay, so negatively charged are groups aspartate and glutamate. So let's say I accidentally put, instead of aspartate, I put glutamate in. Well, it might not affect the overall bonding and therefore the shape of the molecule if those are still going to bond the same way. All right, we back this up here again. What causes a protein to fold in the way that it does is the interactions between, here we go. All right, so, all right, this is my primary structure, my amino acid sequence. the way that the R groups and the amino acids, the way that they are bonding to each other is what causes the entire 3D shape to form, either into an alpha helix and a beta pleated sheet, and then those fold up even more and even more. So we know that proteins are very 3D shape specific, but if the amino acid that I'm replacing this one with by accident is polar and or non-polar and it's still in that same category, then it might not affect the overall outcome. All right. So it might not have an overall effect on the protein shape or function. Another thing to think about, for example, enzymes. Enzymes can be obviously we know enzymes are very important. They're in charge of all of the chemical reactions, which is in a human body, you have 37,000 billion reactions, 37,000 billion reactions happening every second in your body, every second. And most of them are mediated by enzymes. Okay. So enzymes we know are very important. And if we denature them, then we can have an overall failure of our system and cause death or, severe illness. But what we have found is that mutations of an enzyme really are only affected by the amino acids that are in the active site. If you remember, the active site of an enzyme is the area where the substrate is binding. Okay. So if the amino acids that are in this 3D shape, all right, so for example, if we look at this picture here of catalase, all right, so catalase has these, the active site here, okay, it's all these other amino acids are obviously important, but the substrate is a molecule of water. It only has to bind in this little area, the active site. If the amino acids in this area are messed up, then the enzyme won't work. But what we found is if the amino acids in these other areas that don't bind to the water are okay, or are mutated or a different sequence, then it doesn't necessarily affect the overall success of that enzyme. Okay. So genetic variation doesn't always produce fitness variation. And that's something that is a more complicated view of evolution. We don't always talk about it because it's more complicated to think about, well, if you have varied DNA, of course you would be different. Yes and no. There are some things that are just built into our being, our being human, that they're not really going to change. All right. So let's look at these pseudogenes a little bit more. Okay. So pseudogenes are genes that are no longer expressed, but whose sequences resemble those of genes that are expressed. And they are, again, the result of a duplication event. So the original gene probably still being expressed, but it's become so different from the original gene that it became duplicated from. Because they aren't transcribed, they don't have, the transcription part of protein synthesis ever happen for them. All of their nucleotides are very free to diverge at random. And what I mean by diverge is mutate. Okay, they can mutate at random, and there's not going to be a consequence to the organism or to the well-being of that organism. So they will eventually be eroded by mutation because they're not at all repaired by natural selection. Meaning, natural selection is never going to select for a trait that that does not have any consequence. Okay, so they don't get fixed, they don't get repaired by natural selection. And they used to be called, pseudogenes used to be called junk DNA, and sometimes we still call them junk DNA, which is a lot of our DNA actually, but some of it actually does make regulatory RNA molecules. Others are used as sites and signaling pathways. So we're more reluctant to use the term junk DNA anymore. A lot of times now they're called pseudogenes. And another example or another thing to think about here with pseudogenes, some of them are not even, their origin is not even human. Okay. Viruses, as we should know, insert DNA into host DNA. But if they insert their DNA into pseudogenes, those viruses never get expressed and they die with the host, which a lot of our genes are pseudogenes. So a lot of viruses have been introduced into our genome and our parents' genome and our parents' parents' genome, that, they is never expressed, so it actually just dies with the host. Did we just solve COVID? I wish we solved COVID. But I, maybe if COVID had been inserted into our DNA in a pseudogene, that would be phenomenal. But apparently not. That's apparently not how it works. Um, and I don't know if there's a difference in um, how viruses determine, if they determine, or if it's just at random, what section of DNA that they're going to um, insert into. I actually have to do some research on viruses later today, so maybe I'll find out. But I don't know if that's at random or if that's like a trait that they've evolved. So, but can you imagine the fact that our genome is like riddled with fossil skeletons of viruses? We are carrying around a viral graveyard. Okay. So how much of our genome, I don't know, how much of our genome is viral genome that got inserted into us and then got replicated through DNA replication and then went through, and passed on to our offspring through meiosis? I don't know. Right. But our genome is littered with the viruses of the past. So this is another little, this is not from your textbook, but I liked that it showed a little bit of the history of genetics. It's an abbreviated list of the discoveries for genetics. So we start off with Mendel's genetic theory of inheritance is rediscovered in 1900. Then we're going to talk about one little thing in here in 1908, which is the Hardy-Weinberg equilibrium. Genetic linkage between characters is discovered in 1911. So some genes you usually inherit together. Fisher, who is a quantitative geneticist, discovers or develops quantitative genetics for continuous characters. Mueller, who's another one, demonstrates the physical basis of mutation. Morgan, Hunt Morgan worked with Drosophila melanogaster, the fruit flies, established the chromosomal basis of genetics. So now we're beyond the DNA molecule. Now we're to chromosomal level. Avery, McLeod, and McCarty determined that DNA is the hereditary molecule. Watson and Crick determined the DNA double helix. So we're not, Morgan establishes chromosomal. I kind of said that backwards. Chromosomal is bigger. We still don't know what's in there. That's when Watson and Crick figure it out. All right, we figure out that DNA duplication is worked out, or replication is worked out in 1958. Genetic code is determined, so A's, T's, C's, and G's, and how that is a universal code, 1966. Recombinant DNA allows rapid cloning of DNA, so that's now getting into some biotech, recombinant DNA, 1972. First rapid gene sequencing is performed, 1975. So this is when we start sequencing everything we can get our hands on. This is when we discovered that archaea are actually different than bacteria altogether. They're more closely related to eukarya than they are to bacteria. This was when we would have seen that fungi are more closely related to us than they are to plants. All that stuff is because we started sequencing a bunch of things. Organisms are genetically engineered by the insertion of transposable DNA, 1982. All right, so again, big hit for biotech right there. PCR is used to clone small amounts of DNA. PCR reactions or a polymerase chain reaction it actually uses an enzyme that they discovered in a bacterium that lives in the bottom of the seafloor in hydrothermal vents, and they discovered this bacteria there, and they started doing PCR with it. So that was 1985, this is how we were doing COVID testing in the beginning. For example, we're doing all PCR testing. So we still use this all the time today. This is how you do genetic fingerprinting for crime scene investigation, all sorts of stuff like that. And then in 2000 to 2003, the entire human genome is sequenced. So the genetics, that's a 100-year span. And we went from Mendel to sequencing the entire human genome. in 100 years. And so the question is, what will happen in the next 100 years, which we're already 21 years into. So a population's gene pool. So this is where we're getting into, this is 19.1. This is going to be a little refresher on some words here for you: genotype, phenotype, and alleles. Hopefully you are familiar with those terms. And this is merely a review for you. So a population's gene pool. Think about, now I want you to start the thought here with, because this will help with Hardy Weinberg. You are a combination of all of your alleles. Okay. You have 2 alleles for every gene, for every locus. So a gene is a section of DNA. It's at a locus, or a loci for plural, that specific area on a chromosome. That's where that gene is found. And the alleles that are available at loci are called alleles. So the alleles are the options. Okay, so if you have, let's see, you're a pea plant and you have the gene for height. You have, because they're Mendelian traits, one or the other, you either have the tall allele or the short alleles. All right. If you have the tall alleles, you're going to have, you're either homozygous or heterozygous. The alleles are the options. We usually signify them as like big A, little a, right? Those are the 2 alleles available for the trait A, okay? Diploid organisms have 2 alleles at each genetic locus. Each individual has a different subset of alleles in the gene pool. So we know that, especially for non-Mendelian traits, which are most of our traits, like eye color, there's so many more options than just brown or blue, right? There's so many options. It's a polygenic trait. There's, a rainbow of variety here. So within our population, of human population, even if you just think of within our state, because it's easier for us to travel to see each other, and population takes into account your availability to travel to each other. Just within our small population here, we have such a genetically diverse gene pool because each individual has a different combination of alleles. Okay, so you might have blue eyes, but maybe you have brown hair. You might have blue eyes and maybe you have blonde hair. You might have brown hair, or brown eyes, but you have blonde hair. You might have brown eyes and have red hair. That's just 2 traits. Get into all of the different traits, and the gene pool is insanely varied. All right. Organisms in general, not just talking about humans, vary in the size of their genome and the total number of copies of their genome. All right. So for example, we have some, like ferns. Ferns have like, I want to say, now I'm going to blank here, like 1,000 chromosomes or something. How many chromosomes do ferns have? 1,200, over 1,200 chromosomes in a fern. Okay, so some species have a ton of chromosomes. It does not imply any intelligence or anything like that. Ferns have been around for a really long time, so maybe it's more of a sign of the amount of evolution that they've had going on within their genes. Who knows? Some species, the total number of copies of the genes that we have, we are diploid, but not all organisms are diploid. Some species of fish and lizards consist of only females that are triploid, and they have actually 3 haploid genomes. That's a very interesting thing. All right, so there's all sorts of variation in that. Evolution of populations is going to be described in terms of allele frequency, phenotypic frequency, genotypic frequency. Okay, so all those Punnett squares you did back in the day and had to assess them, what's the genotypic frequency of it, the phenotypic frequency of it, now it's going to come into play. So genotype frequency, let's look at this. the genotypes are usually what we refer to as like the letters, okay? The letters that you see, the actual combination. We can't see genotype, so genotype can be a little bit tricky. Um, so the proportion of a particular genotype in a population is what the genotype frequency is. How many of our population have this genotype? And for all these frequencies, we're going to express them as a decimal fraction. So they're actually underneath, they're under 1, because all of the genotype frequencies together add up to 1. So for example, if we think of this title or the total here, like we're adding, okay, let's say in our population of 1,000 or whatever we have, I don't know how many this is, 1,000, yeah, right there. Let's say 490 of them have the genotype big A, big A. We would do 490 over 1,000, and that would give us 0.49. And that's what our genotypic frequency is. Let's say of our population here, big A, little a, we have 420 of them. So we would do that over 1,000 and get 0.42. And then little a, let's say we only have 90 of them, it would be 0.09, okay? So it should all add up to be 1. That's one of the ways we self-check ourselves here, okay? The proportion phenotypic frequency, though, is going to be a little bit easier to see because we can see the phenotype. The phenotype is the physical trait. So it's the proportion of a particular phenotype in the population. Okay, so phenotype, we either have the dominant phenotype or the recessive phenotype. So let's say we're talking about plant height in peas. Tall is dominant over short, which is recessive. Okay, so if two alleles are dominant and recessive, if the two alleles are dominant and recessive, the dominant phenotype is the sum of two genotypes, big A, and big A, little a. So we don't need to know that to get to write these down, but it's important to remember this. So let's say 910 of our pea plants are tall out of 1,000, that's 0.91. Let's say 90 are short, that's recessive, that's 0.09, okay? But what this means for genotypic frequency is that 910 of them have at least one big A, okay? So we'll talk about that. So that's phenotypic frequency. Now you have to calculate allele frequency, and that's where it gets a little bit trickier, all right? So I think the easiest thing to calculate is the one we just did. If you're told in mice, white fur is recessive to black fur, and I tell you the recessive, there's 90 white mice, well, then you know, and you know your population size is 1,000, you can do 1,000 minus 90 and get 910, okay? But how do you know which of these 910 are big A, and which are big A, little a? Which are homozygous, which are heterozygous? That's going to be a little trickier. You know that 90 of them right off the bat are little a, because they're showing the recessive phenotype. So phenotypes, genotypes, phenotype, I think, is the easiest one to look at. The allele frequency gets a little tricky, okay? So because we have to remember that every individual has two alleles, okay? So in a population of 1,000 individuals, we have 2,000 alleles. We have double the amount of individuals. That's how many alleles we have, because each individual is either big A, little a, little a, and each letter is an allele, okay? So when we're looking at allele frequency, we're looking at the proportion of a specific allele in the particular population. So in our population, now you see the total is 2,000, okay? Again, the frequencies still add up to 1. So we know that if I said, okay, we have little a, I don't know if they carried through with this same thing, but if I know that 600 individuals have uh, little a, that could be a combination of the heterozygous that have little a and homozygous recessive. Same thing for big A. Okay. You must count each allele. So if you are given big A, big A, big A's have two big A alleles. Uh, big A, little a have one big A allele, and little a have zero big A alleles. All right. So we'll talk more about this, and maybe this will make a little bit more sense. So Hardy-Weinberg is what we're talking about here. So just to another little tidbit of history is that statistics, so prob and stats, statistics actually have their historical origins in the work of early evolutionists. So a math class was actually founded in a way of studying a science. Okay, so early evolutionists like Francis Galton, Carl Pearson, and R.A. Fisher are three notable scientists who were trying to quantify characteristics like intelligence. So remember me saying that's not easy to do, and they tried really really hard and came up with some interesting things. And this gave rise to an entire field of genetics called quantitative genetics, which I have to say I think is my least favorite form of genetics or study of genetics because it's very math heavy. And I always liked math, but I feel like a lot of my students don't like math, so I don't like torturing you with some math. But the important thing to remember here is that phenotypes, which we can label as just big P in this little scenario here, not for, you don't need to know this for anything further, but just looking at this, the phenotypes of one generation undergo selection and other processes. So the population undergoes selection as well as predation, um, things like that occur, some environmental issues, and the phenotypes determine the genotypes of the next generation. So really think about that. The phenotypes of this generation determine the genotypes of the next generation, because whatever succeeds through selection and the other processes that occur through whichever one is more desirable, through mating, whichever one survives the best, whichever one eats the best and has more children, whichever one just randomly has more children, because if your parents only have one child, evolutionarily they are just as successful as any other parent who has one child. Okay, so sometimes your traits and everything don't matter. If you it just depends on your offspring number. All right, so whatever makes you more fit, that is what will determine which genes get passed on. So the genes of the population predict that generation's phenotypes. Those phenotypes, whichever ones are best and passed on more, determine the genotypes of the offspring's generation, which genes determine physical traits and phenotypes. Generation 2, same thing. They're going to go through natural selection and some processes. So that's going to determine which traits get passed on to generation 3. Generation 3's phenotypes are determined by their genes, blah, 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 continues down, so on and so on. Okay, so genotypes and phenotypes are linked here. All right, so the Hardy-Weinberg principles, it was developed by two guys, Hardy and Weinberg. And so the frequencies of alleles and genotypes in a population do not change from one generation to the next unless influenced by outside factors. Now, we'll talk about the rules that a population must be in to have Hardy-Weinberg equilibrium, as we call it. But the one really important thing to remember here is that a population must be large enough with no selection and no difference in reproductive success. So you're gonna look into this a little bit more on Wednesday, but if we have a small population, it is really, really hard to predict what's going to happen in the next generation, because if I only have 10 individuals, okay, their traits, can be a little bit varied, but there's not obviously not as much variation as if we had a larger population. Whichever one of them happened to have more children, that's going to determine gene generation two, the genotypes, which determines their phenotypes. So let's say you have somebody that's extremely recessive and you would normally think you wouldn't see that at trait as much. For example, cheetahs. Cheetahs are a really cool evolutionary piece here. And this is an example for bottleneck effect. Cheetahs have gone through a lot of environmental, um, issues over the years, and their entire population, they're almost completely homozygous for many, many traits, especially for their immune system. So they, you can take, so think of immune system as, think of like a skin graft. If I were to take a skin graft from one person, or like a transplant, if I were to take a heart transplant from a heart from somebody and put it in somebody else, they would have to be on an immunosuppressant for the rest of their lives, because your immune system will attack those cells of that heart that's not yours. Okay, so our immune system attacks non-self. Well, cheetahs, you can take a skin graft from one cheetah and put it on almost any other cheetah in the world, and it will recognize that skin as self. Okay, because they probably went through a bottleneck in the last few thousand years, and meaning that for some reason something happened, there was some sort of environmental condition, whatever happened, and the ones that just randomly survived were very homozygous for recessive traits. So now the immune system was one of those recessive traits that it didn't attack non-self. Actually, not the best trait, really. And now they all have that. Okay, so you can skin graft any cheetah and it's not going to reject the skin. So that's an example of, why when you have a small population like an endangered species, Hardy-Weinberg is just not going to be applicable. You need it to be a large population where you have a lot more cushion with fluxes and things that can happen. Now, genetic equilibrium. Okay, what this is what Hardy-Weinberg is dealing with. A population that has no net change, meaning no, something can change. Somebody can, a person that is homozygous recessive for a lot of traits can have a bunch of kids. We might see a little bit of a blip, but it's not a net difference because the population is so large or whatever it is. A population with no net change in allele or genotypic frequency over time is said to be at genetic equilibrium. And if allele frequencies do change over time, over successive generation, evolution is occurring. So what this means is that evolution and genetic equilibrium are antagonists of each other. A population at genetic equilibrium is not evolving. Okay. And you might be saying, well, what the heck? How is a species ever at genetic equilibrium? How's a population ever at genetic equilibrium? And I will say that it is rare when you're talking about a trait that is involved in reproductive success or survival success. However, think of like human height. I could poll the class and get everybody's height, and I could study 20 years of height of my classrooms, well, 10 years for me. And on average, they're probably going to be very similar because I teach the same, age group. I teach this, I teach that. That trait is not necessarily linked with reproductive success. So we might not see it have this big effect. Okay. Now that trait, that population might be at genetic equilibrium for that trait. Okay. So this is one of those reasons we study these neutral traits, because it helps us see some things here. So 1908 is when the Hardy-Weinberg principle came about. So if you remember, going back here, very beginnings, Mendel just rediscovered in 1900. We still didn't know about linkages for a few more years. So you had two scientists here. You had Godfrey Hardy and Wilhelm Weinberg. That's a tongue twister. And they mathematically described the expected frequencies of various genotypes in a population at genetic equilibrium. So they took science and observation and they made it mathy. Okay. And the Hardy-Weinberg principle lets us use phenotype frequencies to calculate the expected genotype frequencies and allele frequencies in sexually reproducing populations. Okay. So we can actually predict what will happen with that population over time based on Hardy-Weinberg. So this picture, again, this is not from your text, but I like that it kind of shows us a Punnett square here and shows us what's happening. So let's take this one step at a time, okay? Start with 3 possible genotypes for the trait big A, okay? You're going to have big A, you're going to have big A, and then you're going to have little a. Those are our 3 possible genotypes. Say these genotypes have frequency big P, big H, big Q. Okay, we'll just call them that. The frequency of big A is P plus H over 2. So let's think about that. The frequency of A is going to be P plus half of big A, little a, which is going to be half of H. So meaning if I'm taking all the genes in our gene pool here and I'm looking at them, all the people that have big A, that's P, they're going to be in there, but only half of the alleles of the people that have big A, little a are big A. So I'm going to do half of H, okay? And we're going to call that little p, all right? And then we're going to say the frequency of Q, or I'm sorry, of little a is Q plus half of the H again. So that's going to be little q, okay? So my frequency of the big allele is little p. The frequency of the little a is little q. Take a second and absorb that. I feel like your heads might be falling off if you have not seen Hardy-Weinberg in a long time, then you might be a bit confused, okay? Half of our heterozygous population goes to each because there's 2 alleles. Big A goes to big A, little a goes to the little a's, all right? Therefore, the frequency of big A gametes is little p. The frequency of little a gametes is q, okay? With random mating, gametes combine by the rules of independent assortment, like cards or coins, okay? Meaning, if I have 52 cards in a deck and I pick 2, the combination of those 2 is going to be different most times, okay? Instead of dealing with 52 combinations, our gametes, meaning each card I pull is which sperm and which egg fertilize, instead of dealing with 52, we're dealing with 8 million combinations. So whichever 2 I get, or 8 million cards, whichever 2 I get is going to be very, very different, okay? So to summarize the different combinations of possible sperm and possible egg, we use a Punnett square, all right? That's what a Punnett square is. So we represent frequencies of alleles and genotypes in the next generation by putting a little prime. So you're going to see that, okay? A little prime next to it, P apostrophe. So let's look at this Punnett square. So these are the sperm and these are the eggs, okay? We have big A and little a, big A and little a. We're taking 2 heterozygotes because this is showing us the most diverse population, or it includes all possible genotypes, okay, and phenotypes. So we're taking 2 heterozygotes and we're looking at them in this Punnett square. If the big A fertilizes, which remember the frequency of big A is little p, so the frequencies are written here. If big A sperm fertilizes big A egg, you get AA, which is little p squared, Okay. If big A sperm fertilizes a little a egg, you get big A, little a. That's a P and a Q. So it's written as PQ. If the little a sperm fertilizes a big A egg, you get big A, little a, which again, PQ. And then if we get uh, little a sperm, little a egg, both of them are Q, so it's Q squared. Okay. So down on the bottom here, I wrote this out. If the Punnett square confuses you, think about it this way. There are 4 diploid genotypes that you could have at this locus. You could have big A, you could have big A, little a, you could have little a, big A, and you could have little a, little a. Their respective frequencies, remember, big A frequency is little p, little a frequency is little q. Their respective frequencies would be little p squared. This one would be little p, little q. I'm not going to say the littles anymore. So p squared, p q, q p, q squared. But we don't keep track of order. So this is p squared plus 2 pq's, because it doesn't matter that they're in a different order. Genotypically, it's going to be the same thing, plus q squared. And that is going to be our equation. All right, so we write this as p squared. It's written out here, but 2pq q squared. All right. And this is going to be our equation here. Okay. So the other thing is, if only 2 alleles, big A and little a, exist at a locus, there's no other option. The sum of their frequencies, so in other words, like not multiple alleles here, okay? If only 2 alleles are there, the sum of their frequencies in a population must equal 1, because if you think about it, that's all of the options available, so it has to equal 1, which is like 100%. So so the first part of our equation here is P plus Q equals 1. This is like 2 equations, and you use 1 to solve the other, so it's like algebra, okay? So little p, remember, is the frequency of the dominant allele, and Q is the frequency of the recessive allele. So the frequency of the dominant allele plus the frequency of the recessive allele has to equal 1. When we know the value of P or Q, we can calculate the other value. We can solve for the other one. So if I'm given if I'm given the frequency of the dominant allele, let's say I'm given big A, the frequency in the population, I would say, okay, well, subtract Q out from both sides. P equals 1 minus Q. And well, that would be if I was given Q, actually. All right, let's say I was given P, I would do it this way. Subtract P out from both sides. All right, so you're mathematically going to manipulate this formula to figure out what the other one is. If I'm given P, I'm going to subtract P from both sides, and I'll get Q equals 1 minus whatever value I was given. And then I'll figure out Q. Okay, so my goal is to always find my P and Q first. All right. Then I can plug my P and my Q into the Hardy-Weinberg equation here, which is, again, that P squared, which is the frequency of my homozygous dominant, plus 2pq, which is the frequency of my heterozygous individuals, and Q squared, which is the frequency of my little a, or my homozygous recessive. P squared plus 2pq plus Q squared equals 1. Okay. Hardy-Weinberg principle states that genotypes in a population at genetic equilibrium occur in this frequency. All right, so these are the 2 equations you need to know. P plus Q equals 1, P squared plus 2pq plus Q squared equals 1. All right, and what they stand for. So Hardy-Weinberg calculations usually begin with determining the frequency of the homozygous recessive genotype, little a, little a. We usually start there because it's the easiest one. Because there's no, if you have this phenotype, you are this genotype. Whereas if you're in, pea plants, if you're tall, well, you could be one of 2 different genotypes. If you're short, you have to be little a. Okay. So let's say, for example, that 90 homozygous recessive individuals exist in a population of 1,000. So the first thing we're going to do is we're going to try and figure this out. We're going to say, okay, so my little a equals Q squared. So Q squared. And they're telling me that it's 90 out of 1,000. Okay. So that equals 0.09, right? So my frequency for little a is 0.09. Now, I know that 0.09 is representative of 2 Qs. So I could find what Q is by doing the square root, okay? So I could say, okay, well, what is the, or actually what you could do, here, let me show you on this, the chart here. I know that looks confusing, but what you're going to do, all right, we solved this one. So we know that little a is 0.09, okay? So we can figure out the frequency of little a by adding, so we're going to figure out some further things here. So we know that the rest of our population must be made up of the big A's and the big A and little a's, okay? So once we figure out, if you do, so you're going to need a calculator. So see if you have the calculator on your phone. You might have to turn yours sideways. If you figure out Q squared, you can figure out Q by doing a square root. I was trying to avoid this because I don't know how to put in a square root, but I'm going to figure it out on the fly here. So if Q squared equals 0.09, then Q must equal, let's see how to do this. I haven't done this in a long time. Square root, oops. Square root, we'll write it like that. Square root, okay, of 0.09. That wasn't as bad as I thought it was going to be. All right, which that is going to be 0.3. All right, so we now know what Q is. Once I know what Q is, I can figure out what P is. Okay, because if I remember, P plus Q equals, P plus Q equals 1. Okay, so I'm going to say, what did I say this was? Okay, so I know that 1, so I'm going to rearrange here. I know that Q is 0.3, so I'm going to say P equals 1 minus 0.3. So P equals 0.7. Okay, now I'm going to figure out my other things. All right, so let's see here. If I have my P and my Q, I can plug in my equation here, okay? So I can say, hold on a second here. I can say, okay, P, remember my second equation, P squared plus 2PQ plus Q squared, okay? So P squared, easy, all right? So 0.7 squared, 0.7. Now you can use an exponent, or you can just do 0.7 times 0.7, gives you 0.49. What that represents is all of the big A's in my population, okay? Now I can figure out the percent or the frequency of 2PQs, which are my heterozygous, by doing 2 times, so 2, let's put this in parentheses in case you will look back at this, 2 times 0.7 times 0.3. So it's going to be 2 times 0.7 times 0.3, and that's going to equal 0.42. All right, so we have that plus q squared, which we already know is 0.09. Oops, 0.9. Okay, because we had that up here. So that should all equal to 1. Now, what you can do is you can always double check yourself. It should be close to 1 plus 9. Oops, 0.49. I wish I had a calculator on my thing here. 0.49 plus 0.42 plus 0.09 equals 1. Okay, so we're good. So remember, this is showing us, this one's showing us big A, big A. This one's showing us big A, little a. This one's showing us little a, the frequencies of each. Okay. All right, so now we're going to look and see what this looks like here. So we know that the frequency of big A was 0.49. We know that the frequency of little a was 0.09. Okay. And we figured out that the frequency of big A was 0.42. Okay, right here. So the frequency of alleles, now this is where it gets trickier, okay? Because if you have, let me make a new page here. We have our frequencies of these. Let me just put it over on the side. Okay, so now that we have our p squared, 2pq, and q squared, we can figure out frequency of A and little a. Okay, so everybody that has a big A is going to have 2 copies of big A. Okay, so you're going to have, well, what we're going to do is, well, think about it this way. This guy, only half of these ones have a big A. Okay, so you're going to say 0.49 for our big A category. All right, only half of our heterozygous have them. So it's going to be half, so 0.42 divided by 2. Okay, and then the same thing for our little a's. All right, only half, so it's going to be 0.09 plus only half, 0.42 divided by 2. All right, so that is what this 0.21 is. Okay, we took the 0.42 and we divide it by 2. So what you get, you're double checking, is 0.49 plus 2.1 gives you 0.7. Same thing here, 0.3. Now we already did this up here. Okay, just want to double check and make sure that our things add up. All right, so this is 0.49 plus 0.21. Okay, that's going to give us 0.7, which equals P. And this one is giving us 0.09 plus 0.21 equals 0.3, which equals Q. All right. So I know that this is a lot of mathy stuff, but it makes sense. You're just algebraically plugging things in and assigning where they go. So if we look at what this means in terms of the Punnett square, we know that the frequency of, in the population, looking at the offspring, all right, so we know what the original population was, so we should be able to figure out what the offspring could be. So if we know that the possibility of having a big A is 0.7 and the possibility of having a little a is 0.3, all right, we put those on both sides. Look at what you get. You get your Punnett square. So some people I've seen actually fill it in like this. If this helps you think of it, then do it this way. If you're given the P and the Q and you're trying to figure out what your options are, instead of writing it out like this, some people actually write it in a Punnett square. They say, okay, well, if A and big A are together, that's P, and P, that's P squared, that's 0.7 times 0.7, okay, that's 0.49. Okay, Q and P, so big A and little a come together. That's going to be 0.7 times 0.3, which equals 0.21. Same thing here, 0.21. I would add these two together. Obviously, they're the same color. That's why they're marked that way. And then little a, 0.09. All right. So this is another representation of how to show it. And I think that for some people, that makes more sense. Right. This is showing you everything you need. This is showing you P, showing you Q. So it's showing you allele frequencies, and it's showing you phenotypic frequencies or genotypic frequencies. All right. So let's take a look at this other one here. Give your mind a little bit of a break. See if it will open. No. Right click or control click. Okay.
Imagine a butterfly population with a pair of alleles that determine wing color as shown. We will represent the frequency of the dominant allele as P and the recessive allele as Q. Because there are just two alleles, their frequencies must sum to 1. The Hardy-Weinberg rule describes what happens if a population is at genetic equilibrium, if it is not evolving. In this case, allele frequencies remain constant between generations. Genotype frequencies can be calculated from the allele frequencies, and vice versa. Suppose we observe that the frequency of white butterflies in our population is 9%. If Q squared is 0.09, then Q must be 0.3. Because P and Q sum to 1, P must be 0.7. We can calculate that the expected frequency of dark blue butterflies is 0.7 squared, or 0.49. The expected frequency of light blue butterflies is 2 times 0.7 times 0.3, or 0.42. If the genotype frequencies in a population differ from those predicted by the Hardy-Weinberg rule, this shows that the population is not at equilibrium. In other words, the population is evolving.
Okay. So what you'd have to think about is that this represents a population that is at an ideal situation. And that is seldomly going to occur in the natural world, okay? Because let's face it, nothing is ideal in nature. Things change all the time. If we didn't have our creature comforts of having a house and a car and all these things, we would be living in a very different world, which our ancestors, hundreds of thousands of years ago did have to live through. So this equilibrium thing, it's almost like in physics when you're learning physics and you're everything is like, well, in a vacuum. It's like this is kind of like evolution in a vacuum, right? So genetic equilibrium exists only when 5 conditions are met. All right, random mating, meaning there's no preferences in mating. There's no reproductive success differential. There's no you are stronger, you are prettier, you are, sing a better song. There's none of that. It's random mating. So plants have random mating. They can't control who their pollen and mate with, right? Or yeah. You also have to have no net mutations, which I know sounds ridiculous because mutations are always happening. But think about it more so like in that specific trait, okay? So if it's a trait that usually does not mutate, and we'll talk more about mutations, then okay, there's a chance there, but mutations can always happen. A large population size, like I said, if you have a small population size, you're going to see a bottleneck, and you're not going to be able to see any close resemblance of Hardy-Weinberg at all. All right, there has to be no migration, meaning in or out, so no emigration and no immigration, because then you take your genes with you. All right, so you would be leaving the gene pool, which would leave a void that could be filled in by somebody that did not represent your exact genetic combinations, your phenotype. And there has to be no natural selection, meaning everybody's like living in this perfect world. All right, so obviously most populations are not at genetic equilibrium, okay? Most populations are evolving. All right, so if we look at another example, it's blood groups. Let's say M and N blood groups. We think about A, B, and O. A and B are codominant. We're not going to talk about O because that's just a whole other ball game. But just looking at our codominance alleles for blood typing for humans. So let's say you're going to have 3 different phenotypes here, okay? As opposed to before when we had only 2 phenotypes, we had blue and white butterflies, okay? This example is codominance, so your heterozygous is going to have a different phenotype than either of your homozygous dominant or your homozygous recessive. All right, so the genotype big M produces only the M antigen on the exterior of the red blood cells. The genotype NN, sorry, dropped my ring because I'm playing with it again, produces only the N antigen. But the heterozygous genotype produces both the M antigen and the N antigen. Population geneticists can directly observe all 3 possible genotype frequencies and compare them with calculated frequencies. So they can actually see if the population is at genetic equilibrium, because they know what genetic equilibrium should be, and they can take people's blood type or take blood samples and identify if this population is at genetic equilibrium by comparing them to the expected results. So let's take a look. Let's say that our big M genotype, our observed, is 320 out of 1,000. Well, that we know is P squared. All right, so I'm just going to put P squared in here. That's what that means. Okay, we know that, I don't know why these are underlined. I think this one's to show it's like you're adding up, but MN is going to be our 2 PQs. All right, and then big N, or NN is going to be our Q squared. And we're given those values, okay? So what this is, let me change this underline here. And we know that our population has 1,000, all right? So the frequency of M alleles is going to be, now remember, count them up. Everybody that's got this has 2 of them. So the frequency of M alleles, which is P, is 2 times 320, okay? Because each 320 people have 2 big M's. So if you're given the amount of them in the population, you have to multiply it by 2. So if you're given, this is like a good rule here. So if you are given the number of individuals in the population with each genotype, to find P, multiply the homozygous dominant, I'm going to say the number of homozygous dominant individuals by 2, and add it to the number of heterozygous individuals. All right. Because here you see, I didn't calculate the frequency. So this is a different problem than here. Okay. Up here, to find P and Q, we didn't have to multiply them by 2 because we were already working with a decimal number that we had already calculated what that frequency was. Here, we are working with a number of individuals out of our population. Okay. So I would say, all right, well, P is going to be 2 times 320, because that's 1, 2. And then MN only have 1. So I don't have to multiply that by 2. I can just add that. So 2 times 320 plus 480. That's going to give me, let's do our little math here. 2 times 320 plus 480 gives me 1,020. 1,020, divide that by 2,000, and I'm going to get 0.56. okay? Then I'm going to do the same thing. So we're going to say, do the same thing for homozygous recessive, okay? So for my homozygous recessive, well, I know that Q squared, or big N is 200. You know what? I'm going to delete these because this is actually, that would be giving the frequency. So if you wrote that down, delete that. This is just giving us the number of individuals. So I'm going to say, okay, big N is 200. So 200 times 2 would be 400, plus all of these individuals would be 480. So I'm going to be 880 divided by my 2000, and it's going to be 0.44. Okay. Does anybody have any questions? See, this is why I'm only going to now slide 39. I added those pictures in because I don't want to hurt your brains that badly. Hopefully, if you are like, oh, this is so easy, yay. Usually I do not get the response for Hardy-Weinberg. So I am aware that it's difficult. So do not feel bad if you're confused. I think the practice problems will definitely help. All right. So if this population is in genetic equilibrium, then you can figure out your P squared, 2PQ, and Q squared. Okay. You'd figure out, we know P, we know Q. Square your P. That gives you how many people should be MM. Okay, 313. You want to know how many heterozygotes you should have? Again, this is like if I do my, now there is a jump here. They figure out P squared and then they multiply that by the 2000 to figure out how many individuals it is. Okay, so once you find P squared, you'd multiply it by your population number, which is 2000, to figure out how many individuals should have that genotype, 313, okay? So if you do 0.313 times 2000, it should be that. Why does that not sound right though? 0.313 times 2000. Yeah, hold on. What am I doing wrong here? Oh, I guess our population is only 1,000. Never mind. So then when they sampled it, they only took 1,000 individuals for their sample. All right. So you're just going to multiply it by your population size and get what you should expect. Okay. So let's see, where was I? Then you would expect 0.492 for 2PQ and 0.193 for Q squared. So then you would compare your observed, which we already had, to your expected. Okay. Now, in order to do that, you would have to do something called chi-square analysis. Okay. So let me, I'm going to show you based on our examples here. So chi-squared, don't ignore this for right now. Let me delete that. Chi-squared is when you are going to compare your expected results to your, so compare observed results to expected results to see if there is a statistically significant difference or if any difference is just due to chance. Okay, so if there is a statistical difference in this kind of example, then that means, so if a statistical difference is present, then your population is not at equilibrium or there was an experimental error. All right, usually in this situation, it usually means you're not at equilibrium. So let's look at what this thing is. This is called the chi-square test. So this little x, it's not actually an x, it's a chi, but Greek letter. But chi squared is going to be the sum of all of our observed and expected results. Okay. Now we have 3 sets of options. We had big M, big M, big N, little, or I'm sorry, MM, MN, and N. We had 3 options. Okay. I'm going to just copy this chart onto our slide here. Whoops. Copy, paste, there we go. All right, so these were our observed results, okay? And we know, I'm gonna insert a column here, and we're gonna say these were expected, and we're gonna take those from the slide right above. We expected 313 individuals to have that. We expected 492 individuals to be MN. And we expected 193 individuals to be NN. Okay. So that should, now when I worked this out ahead of time, let me see if we get an exact number here, 313 plus 492 plus 193. Yeah, this actually gives me 998, which is important. All right. So for whatever reason with our analysis here with rounding, we got a little bit of a difference here. All right, so we'll just make sure we keep that into account. Consider that. All right, so for our chi-squared here, what you're going to be doing is you're going to say, okay, now this is a little bit trickier because it would be easier if I had like a thing I could write on, like some sort of tablet where I could write insert formula. I don't think they have that on slides. So we're just gonna go with here. All right. So my chi-squared, and I'm gonna just write it as an x. All right, chi-squared is gonna be equal to, all right, the first one. So MM. Okay. Our expected. Come on, cursor. There we go. Our expected or our observed was 320 minus what we expected, which was 313. And I'm going to square that. And we divide that by our expected, which was 313. Okay, so that's my first set. Now I'm going to do it for my MNs because this big sigma sign means sum of all of these. Okay. All right, my observed 480 minus my expected, which was 492. I'm going to do that squared. And then I'm going to put it over my expected, which was 492. So that's the MN group. And now my NN group, observed 200 minus expected 193 squared over 193 expected. All right. And then I'm going to add that all together. All right. So we do this again. You might want to turn your calculator sideways if you're using your phone, or I don't know how it is on iPhone, but I get like a scientific calculator when I turn it sideways. So we're going to say, okay, do all this calculation, and you're going to get for your first number, oops, hold on. Shoot. 320, that's 313 squared. 313. Okay, so I got, this is x, 0.157. I'm going to go 3 decimal places plus, sorry if you're already done here, just throwing these calculations in so you can see. Doesn't take you too long. Actually, there's be plus 3 plus. Usually I'd be doing these on the chalkboard, the marker board. Okay, so my chi-squared is Ah. Okay, my chi-squared equals, so then I'm going to add all those up. 0.157 plus 0.293 plus 0.254 is equals 0.704. Okay, now you're going to say, well, what the heck does that give me? All right, that is going to give you your critical value. All right, so this value then, you are going to use this value to look at your chi-square chart and use the column for 0.05, which means 95% certainty. Now, this is when you guys study or you do any type of, if you read any type of scientific journal article, all of results, when you do a scientific study, all of your results have to withstand chi-test analysis. Okay, so this is chi-square. All right, so this is the chi-square chart. So now you might ask yourself, what, hold on a second, this was for my other one. So you might ask yourself, what the heck is this? All right, this is showing us using two different things. We're using our percent of our certainty. So we're always going to use the one that says 0.05 because that means we're saying with 95% certainty that if our value comes into this chart at the right place and it's less than this value, then our population's at equilibrium or our results are valid. They're statistically valid. Okay. If we come into this chart and our value that we got is above what we're looking at, then we know that our population is not at equilibrium or our values, we did something wrong with our experiment. Okay, so chi-square tests are something that all scientists have to use, which is why I want to introduce you to it. All right, so if we look here, you're going to see degrees of freedom on the left side. This chart is the same, just split in half. So like degrees of freedom continues over here. All right, so degrees of freedom, how you measure that is by the number of options, right? Degrees of freedom, which is lowercase d. Oops. All right. Is the number of outcomes minus 1. Okay. So, for example, we had 3 genotypic options. So 3 minus 1 equals 2. So use the row where D equals 2, okay? And we are use the column that is 0.05, which would be 95% certainty. All right, so, okay, we know we have D equals 2 and this right here. So we want to compare our number that we just got to that one right there. All right, so for our example, what was our results here? 0.704. 0.704 is less than 5.991. So our population is at equilibrium. And we had no experimental errors. All right. Now I did include in here the next problem, which if I just copy this chart here, you can put this is an example of one that is not going to be at equilibrium. All right. So with this one, we had 330 there, we had 512, and we had 156 observed. Okay. So our values, we wind up with a critical value of 0.82. All right. Or I'm sorry, 8.82. So for our chart here, it is way higher than 5.991. So we know that our population must not be at a Hardy-Weinberg equilibrium, or we have some sort of experimental error. So I did want to include an example where there was an error. Okay. All right. Any questions on chi-square test? This is an important, I know it's annoying. Trust me, when I was learning it, I hated it. But it's really useful when you're doing scientific journal articles, you're looking over them, reading them, analyzing them for a class. You have to be able to know if your data is statistically significant. If you've ever seen statistical significance, this is what that's looking at. All right. Okay. If you have questions, just let me know. All right. So the last few little things here is we're going to look at these mutations and some inbreeding stuff. All right, so this term microevolution, okay? Microevolution is happening at a small scale, all right? Think of, I think we've touched on this before, but microevolution refers to a relatively small generation to generation changes in those allele and genotype frequencies within a population, all right? So there are 5 microevolutionary processes, the opposite of conditions for genetic equilibrium. And these things will cause our populations to not be in Hardy-Weinberg equilibrium. The first one is non-random mating. Okay, so we'll talk about that. The second is mutation. All right. Now, those are the 2 we're going to talk about today. We're not going to talk about genetic drift, gene flow, and natural selection until Wednesday. All right. So let's look at non-random mating. One example is inbreeding. Okay. Inbreeding is the mating of individuals who are more closely related than if they had been chosen at random from the population. In order to be technically inbred, you have to have shared genes with at least one relative. Okay. Now, the problem with inbreeding is that it increases the frequency of homozygous genotypes, and it reduces, therefore, heterozygosity, which in turn decreases genetic variance. Okay, so now inbreeding in biological terms is different than in societal terms, because like in society, marrying your second cousin apparently is not incest, or your third cousin. There's like I can't remember which one it is. But in terms of genetics, that's inbreeding. So like when we study certain populations that are very isolated, even like studying the Amish and Mennonite populations, we see a lot of this inbreeding. Studying royal families, we see a lot of it as well. And it might be legal, it might be not frowned upon societally, but if they have genes from the same ancestor, then that is considered inbreeding. So many diseases are recessive. Okay, we know that. And if inbreeding increases the frequency of homozygous genotypes, then it could increase the likelihood of a recessive homozygous genotype. Okay, meaning you would have that disease. Statistically, this is interesting. Statistically, we are unlikely to mate with an individual carrying the same disease alleles if we're not related to each other. So in nature, if you have the carrier for a disease and another carrier for a disease, you're statistically less likely to actually mate, right? So inbreeding may cause inbreeding depression, which is when inbred individuals have lower fitness than those that are not inbred. So it can cause definite depression in your likelihood of, or your fitness level, okay? An example would be inbred white-footed mice. They have a decreased survivorship Now, this is the data your book gives you, which I think is really dumb because it's kind of very incomplete. But this is showing weekly survival rates of field captured mice, non-inbred and inbred. Um, so they captured the mice, they were marked, and then they were released back into the field population. Here's our little description here. The population was then sampled by capturing mice, noting the number of inbred and um, non-inbred mice, and re-releasing them back out. All right, the y-axis represents the 10-week period during which the samples were collected, but it's missing weeks 4 through 7. Values on the y-axis are the estimated proportion of mice that survived since the last time the population was sampled. For example, the value of 0.2 at week 3 for the inbred mice means that 20% of the inbred mice that were alive at the beginning of the week survived through that week. All right, a lot of capturing of mice, which doing studies on mice is actually interesting because you lose a lot of them while you're trying to weigh them and do all that kind of stuff. But this, they were just using survival, so that's a little bit easier to quantify. So if you look here, we see the non-inbred mice definitely are always statistically higher than the inbred mice. So this would be the inbred mice are suffering from an inbred depression. All right. So do we have inbreeding in nature? The answer is, yes, somewhat. So it's common on farms and in scientific labs. All right. We do these, or in certain populations. Small populations lead to inbreeding. So, very isolated cultures. Small cave invertebrates, self-pollinating plants like peas, plants with very local pollination, meaning, you've got, I think of like these little micro gardens in the city. There's like a little garden inside of a big concrete city. Like, there's going to be very local pollination there. And selfing worms like nematodes, they actually all have high levels of inbreeding. And that can actually lead to accidental differentiation of local subpopulations where they can form into very different like bimorphism where they start to actually diverge very much so and can lead to speciation. But outbreeding, which is the opposite of inbreeding, is going to be much more common. It's extremely common. For example, a fruit fly, the famous Drosophila melanogaster, our famous fruit fly we use for so many studies, they don't fly as they don't fly very well. They don't walk very far, but they exchange genes over hundreds of square miles. A little tiny fruit fly, not a house fly, a fruit fly, hundreds of square miles. Pollen can spread surprising distances. All right, we can see pollen grains travel very far. Birds fly, animals migrate. So most animals outbreed, but we do see inbreeding in self-pollination and some invertebrates. So there are actually also some good reasons in agriculture for inbreeding. Genetic polymorphism, which is where you're going to have many different phenotypes of outbred populations, is lost if inbreeding is sustained. So inbred lines fix single alleles. And remember, the term fixation in genetics refers to 100% frequency. So you're going to go from where there's not a lot in the population to where there are 100% frequency for that allele. So when you have inbreeding and you have a small population, you're going to have this genetic polymorphism get bred right out. All right, so characteristics that are true to breed often are as a result of inbreeding. So think of breeds of dogs or farm animals, fruits and vegetables that we eat. If you have a Labrador Retriever, you got that Labrador Retriever because you have traits that are true to breed. Dogs are one of the best examples of inbreeding, which is why we have so many different breeds. But plants like fruits and vegetables that we eat as well. When you have, I grow a lot of heirloom tomatoes and heirloom cucumbers and stuff like that. They're not, they're very good and I like them, but they're not as hardy a lot of times as a hybrid. They have true to breed characteristics, but they're not as hardy because they are inbred. All right. So many breeds have genetic disorders from inbreeding. Think of dogs. German Shepherds, St. Bernards, they're known for once they get to a certain age, developing hip dysplasia. inappropriate aggressiveness. Think of, pit bulls. Um, uh, I'm blanking, but you guys know what I'm talking about. Oh my gosh, I'm blanking. I'm such a dog person. I apologize. Um, we think of them as aggressive. That was because that was bred for them. They are actually, it's not their fault that they're like that. It's actually our fault. And we also can't deny the fact that we did that. So I love, I know many of absolutely friendly and lovable pit bull. At the same time, they're more likely to have a bout of inappropriate aggressiveness than another breed of dog, because that is a true to breed trait that we have bred into them. It's not obviously not guaranteed, but it's just much more, the statistics are higher. Blindness, I think it's Dalmatians are much more likely to have to suffer from blindness. So these are true to breed characteristics that are unfortunately side effects of inbreeding. And there is something called hybrid vigor, which is where you cross two unrelated inbred lines and the offspring actually thrive. And a lot of times these genetic disorders are bred out of them a little bit. So for example, this hip dysplasia, like a Bernedoodle. Okay, a Bernedoodle is less likely, which is that one's a Burmese mountain and a poodle, but it's less likely to have some of these side effects that a straightforward Bernese mountain dog and a poodle would have, because they have the hybrid vigor. All right, that's a term, that's an actual term. All right. So another form of non-random mating is called assortative mating. And assortative mating is where individuals select their mates by phenotype. So this is the more common one that we think of with like cardinals or birds, I should say. I'm just thinking of cardinals because we had so many with our bird seed feeders. So assortative mating is, one mate is preferred over another based on bird song, based on color, based on feather pattern, based on antler size, based on whatever it may be. So this is going to change the genotypic frequencies only at the loci that are involved in mate choice. So we can study the other genes, and those genes might be at equilibrium. However, the genes for this trait that might be making you have a brighter red feather if you're a cardinal, that one is going to be out of equilibrium, because that's involved in, it's assortative mating. It's non-random. Selection of mates with the same phenotype is called positive assortative mating. And selection of mates with opposite phenotypes is called negative assortative mating. So this negative assortative mating can lead to where you have two very distinct phenotypes for males and females, because you choose the one that looks very different than you, which is usually the other sex, having that trait. All right. The second piece that we said can lead to this microevolution thing, this mutation. So to knock some population out of genetic equilibrium. So we all know a little bit about mutations, right? A mutation is a change in DNA. You should remember there's duplication, deletion, insertion, frame shift mutations. Maybe all that stuff's coming back to you. All right, but there's a little bit more to mutations than meets the eye. The source of a genetic variation in a population is a mutation. So like we said, mutation is the raw material for natural selection to act on. Only mutations in reproductive cells are inherited, as opposed to what Jean-Baptiste Lamarck thought, because we didn't know yet. When a polypeptide is sufficiently altered to change its function, the mutation is usually harmful. All right. Different allele sequences sometimes specify different amino acids, or they may lead to increases or decreases in the total number of a particular protein, or it could change when that protein is made. All right. So mutations that cause a polypeptide to be sufficiently altered are usually not good. Some things are just not going to mutate. All right. And this is, if you think about it, some things are in our phenotype, our whole organism phenotype, are ancient, stable, and have what we call developmental buffering mechanisms to keep them that way. And so they resist the mutation of phenotypes. So for example, we have 4 limbs. That doesn't change very frequently at all. It is extremely rare that you would find somebody that has an extra limb. And usually that is actually a twin situation where, there's some sort of something like that. Our four limb mechanism is so ancient and so stable that it has a developmental buffer system built in. All right. Genes which get buffered are actually more likely to develop neutral mutations because the effect of those mutations is buffered out. So it's kind of like, well, you screwed up, but it doesn't matter. We're just going to do it anyway. All right. They're not exactly sure why this happens. It's called canalization, but it allows for hidden genetic variation to actually accumulate in really important genes because of this buffering system. Again, this is still very cutting edge new stuff that we don't 100% know what's going on there. But some mutations are just much less likely to occur. Some mutations are not random. All right, we think of mutations as a random change in DNA. However, some mutations are not. Some sites are more frequently mutated than others. So some genes, we think of breast cancer. Breast cancer, we know BRCA1 gene, BRCA2 gene. There's links there. Some genes are more likely to mutate, right? An example of another thing that some mutations are not random, pathogenic bacteria, bacteria that causes illness, okay, like salmonella. In a challenging environment, pathogenic bacteria will actually downregulate DNA repair after their DNA replication. DNA replication you have, and then you have enzyme, DNA polymerase comes in there and repairs some things that it did, just in case there's mistakes. DNA in bacteria that is pathogenic actually gets, the repair mechanisms get shut off to promote mutations. So pathogenic bacteria actually do not, it's not random. That's like they evolved to promote more mistakes, because that's going to allow them to trick the system a little bit more of wherever they're living, and the challenging environment that they're living in, antibiotic resistance, something like that, antibiotics they're living in or whatever, then there more mutations mean they're more likely to have some that are created that don't have that have a better genetic combination. Mutations or nucleotide classes. So let's say you have an an adenine, and it gets accidentally, there's like some sort of mutation, and adenine is more likely to mutate into a guanine because those are both purines. All right. A thymine is more likely to mutate into or be replaced by thymine is more likely to be replaced by a cytosine because they are both pyrimidines. You're less likely to have an adenine be replaced with a thymine or a cytosine. All right. So nucleotide mutations, they are random, but they're usually within their class. Okay. So that's a little bit less random. And mutations do not produce random change in phenotypic space. Like, for example, you're not going to have a mutation that's going to randomly have a human growing wings. That would be really cool, but it's not going to happen. All right. They don't just create a new phenotype space, meaning like this phenotype never existed before. So we're going to, all of a sudden, we're going to have wings. All right. That would be a very different process. Okay. But mutations are random. Sorry for that grammar. Mutations are random in ways. Okay. This is big. They are not dependent upon the need of the organism or convenience. The needs of a population do not determine what mutations will occur. This is a very common mistake. They happen by chance, and they can affect the offspring if they're in a gamete. I can't tell you how many times I've seen, well, the giraffes got the long neck because it was beneficial for them. And they needed that long neck to eat from the trees. Yeah, I see what you're trying to say, right? But a mutation does not develop because of need. The reason why the giraffe has a long neck is because it had a genetic mutation that caused it to have a slightly longer neck. It can eat more. It passes on its genes to its offspring, and the offspring are more likely to have longer necks then because of genetics, and they can eat more and can pass their genes on. It's not because somebody needed to. And this is really important. We have to take the humanization, we humanize these traits. They are not humanized traits in nature. They are just random, and they are not, dependent upon what that population needs. And so the next section deals with genetic drift. So genetic drift and the random events that change allele frequencies. Genetic drift is going to occur when, just think about it as the genes and the alleles of a gene pool of a population are drifting, okay? And so it's going to be when something's drifting, it's a slow, progressive movement, you're just drifting down the river, whatever it is. Genetic drift is going to be the same kind of thing, all right? So random evolutionary changes in small breeding populations results in changes in allele frequencies in a population. So when we have a small population, a small breeding population, you're going to see the effects of these random changes. You're going to see them a lot more drastically. So what's going to happen is you're going to see a decrease in genetic variation within a population, but an increase in the genetic differences among different populations. So what that means is that it's going to become more isolated as those changes occur and they add up. It's going to cause the population. Hold on, I've got a barking dog situation. Sorry if you can hear that. It's going to cause the population where those changes took place to become very different looking, and they're going to become very isolated from the other populations that are around them. So let me see if I can put this in here. Let me stop the screen share and get it back. I want to show you guys a little, I don't need the background, run experiments. So this is showing us a koi pond, and actually one of your questions in your lab deals with this. This is a really cool simulator that shows how, if we have this population size, let's say we leave everything the way that it is, mortality rate, brood size, how many babies each mama has, the ratio of males to females is 50%. The carrying capacity is the same size as the initial population. There's no migration. So we're at, we would assume based on all of these things, there's genotype relative fitness. No genotype is better than the other. Mutation rate 0. So assortative mating is 0. So we have random mating. So this is going to be a population that we would assume is at genetic equilibrium. So if I look at, as the generations go on here, and let me speed this up, let's look at what happens after 1,000 generations. Look at the genotype proportions as we're going through here. Not 1,000 generations, 100 generations. All right, and the population size is staying pretty consistent. It wobbles a little bit, but it doesn't, get over that 200. The allele proportion is staying pretty consistent here. You might see some dips. So after about, this is fun if you actually look at the fish over here. Oh my goodness, makes you dizzy. If I stop it at 100 generations, and I look at the relative genotypes or relative proportions of these genotypes, what I'm seeing is that they stayed pretty consistent here. All right, they did wobble a little bit, but they stayed fairly consistent. Okay, now let's say I'm going to go back here to my design. All right, and I'm going to say, okay, let's make this a lot lower. Let's make it 25. Okay, so sometimes this thing acts a little bit weird. Stop. Okay, make it 25. And it's not. Hold on a second. This isn't right. Let me reset this. Okay. Now watch these genotype proportions here. Let me speed this up. You're going to see them kind of go all over the place. All right. And you can see it with the fish. We've got a bunch of spotted. Now we've got a lot of white. There's more red popping up. You can see it here. Okay. Because this is a small, and look at the population sizes all over the place because I left that carrying capacity. Now it's going to start to level out. All right. If I go back to design and I make, let me pause this, refresh, I'm going to make the carrying capacity 25. And let's go to the data, restart it. Okay, now you're going to see this allele proportion is going to change as we go through. All right, so this is just a simulation to show us how the smaller the population. Now notice that the homozygous ones, which normally if something's homozygous recessive, we wouldn't expect it to be having, a lot of them in the population, but you see it grows and then it shrinks. Because with a small population, you really just can't predict. Look at this allele proportion, all right? You just can't predict what's going to happen. So in a small population, an allele present at a low frequency could be completely lost by chance, all right? So if we were looking at look at this, okay? We've got the homozygous dominant down to 0.04. That means only 4% of the population, which I don't even see one in here, but very small percentage. It can just be randomly lost if the one who has that trait dies off from a predator or whatever it is. So you get with these, you get a random, what we would call wandering of neutral genes. It actually resembles what's called Brownian movement, which is like as particles of water hit dust, and when you're looking at it in a microscope, it looks like the dust is moving. The dust isn't moving, it's called Brownian movement. But this can happen. And a neutral gene, all right, a neutral gene would be a gene that doesn't impact anything. It doesn't, it's one of those genes that it doesn't code for a trait. It can make it, if it makes it through meiosis and gets into a zygote, all right, the organism is going to grow up, and that person or fish in this example could have a large family or a small family just by chance. So depending on how many kids that organism has or offspring, you might see all of a sudden a lot of these, neutral genes in the population. All right. So if variation plus reproductive success is correlated with a trait or a gene, it will produce natural selection. Meaning, if it makes you more successful at reproducing and you have a variety that makes you more successful, then that's going to lead to a natural selection. But if a genetic variation does not help or does not have any correlation with reproductive success, it causes genetic drift. It just causes the gene pool of that population to drift a little bit. All right. So one example of something that causes genetic drift is called the bottleneck effect. All right. And think of, normally when we think of the bottleneck effect, we think of like an image where you see something like this. All right. If the original population is very varied, and then you allow something happens and only a few survive, whatever those surviving population, whatever those are, that's going to be what the new population will have more of. All right, so a bottleneck, occasionally a population may rapidly or severely decrease due to disease, exploitation like overhunting, or sudden environmental change, like wildfires, things like that. Disease are common. So genetic drift can occur in a small population of survivors. So as the population again increases, now as these guys repopulate, many allele frequencies may differ from those preceding the decline. So let's say, for example, that two diploid organisms survive from a population of 100. Only two survive, whatever happened. They only carry 4 copies of a gene because they're diploid. So one's got big B, the other's got little b, whatever it is. If there were 20 alleles, let's say to begin with, so like, multiple alleles for a trait, which is very common in the original population, only 4 alleles are max get passed down. Think of blood type. We have blood type A, AB, B, O. That we have normal proportions. If all of a sudden there was a meteoroid hit Earth and only 4 people survived and they all happened to be blood type O, well, then everybody's from there is going to be blood type O. All right. An example of this, I think I was telling you guys, cheetahs. They're almost completely homozygous, especially for immune system. You can skin graft from any cheetah to another and it will be recognized as self. So it's assumed that they probably went through some sort of bottleneck event in the last few thousand years. We don't know what exactly. All right, so this is the bottleneck. Now another one, which is very similar, but it's more, think about the name. Instead of them going through a bottleneck, one set of organisms is leaving and going to find a new place to live, the founder effect. We see this a lot with human populations, which is part of the case studies you guys are going to do. So founder effect is genetic drift that results when a few individuals from a large population found a new colony. All right. The only alleles in the new population will be those of the colonizers. So allele frequencies are usually quite different from those parent populations. So it's almost like you'd want to pick the most representative people of your species to go and found the new colony, because otherwise many many phenotypes could be lost. So one example is the Finn population, Finnish people. They are thought to have descended from a small founding population. I think their population now is like 320,000. And they're thought to have descended from a small founding population that remained separate from the rest of Europe for centuries. So they became very isolated. And so that is one of the reasons why they have characteristics that are very, they're like true breeding. They have characteristic pale skin, blonde hair, blue eyes, and it's very common amongst all people from Finland. So the bottleneck effect and the founder effect are different in their source or their how they occur, but the result is very similar. So founder effect is going to be also found in certain genetic diseases that are rare overall, but common in some populations that are founded by small groups of people. In your case studies, you guys are going to look at 3 populations. You're going to look at Tay-Sachs disease in the French-Canadian population of Quebec. Tay-Sachs is also common in Ashkenazi Jew population. So, for example, which is a European Jewish population, but a lot of times, like when you're, let's say, this happened with a friend of mine, her and her husband were trying to have a baby. Actually, no, I think it was when they got married. They had to have, I'm trying to remember, they had to have blood testing. They used to be where if you were going to get married, you had to have your blood tested. And I think that some places still do that. And they were one of them. And probably around here, they don't do it, but they live in central New Jersey and maybe it's a more diverse population. I don't know, but they had to get their blood tested. Turns out that they both were carriers of a certain type of disease that is common in Ashkenazi Jews. And so they had to just make sure that they were aware of that for when they have children. So Tay-Sachs is also, there's a founder effect example of it in Quebec. Porphyria is another really interesting one. It's in the Afrikaner population, which was a population found in South Africa, the country of South Africa. And then diabetes, this one's actually a really interesting story because it involves a story that's been told in movies, and Mutiny of the, or I think it's the Mutiny of the Bounty or something. It's like a famous story, but it led to some founders settling in the Pitcairn Islands. And right now, which these islands are considered like paradise, they're Polynesian islands, but there's a massive obesity problem and the link, and diabetes, type 2 diabetes. And so there's a link to possibly the founder effect being a factor. So you guys are going to tell us about that in the case studies. So gene flow generally, so what gene flow is the movement of alleles. And that generally increases variation within a population. All right. So the migration of breeding individuals between populations, think about it like this. If you had, your textbook uses bullfrogs. If you had little ponds all over and they all had their own little population of bullfrogs, occasionally one bullfrog might hop over to another pond and mate, but there's more likely that the bullfrogs in one immediate area are going to mate and the other ones would mate. And so over time, those populations could actually become a little bit different from each other. But occasionally, if you do have migration of breeding individuals between a population, there's going to be a corresponding movement of alleles, okay, which we call gene flow. And it increases the genetic variability in the recipient population. That pond that the little bullfrog hopped over to now has a whole new set of genes to play with. Right? So gene flow between two populations increases the genetic similarity between those two populations. It's like the opposite of what we would call speciation, where a new species is formed. It's actually securing the similarities between them. And it counteracts the effects of natural selection and genetic drift, which would make the populations more distinct. Genetic drift would say, okay, well, you guys are not mating frequently, so you're going to become different. And the genes and the gene pool in your pond is going to slowly become a little bit more different over time. That's genetic drift. Gene flow, think about it like, okay, I know that these words sound very similar, genetic drift and gene flow. Genetic drift, the genes are slowly, gene pool is shifting over time, unless it's a small population, in which case it can happen pretty quickly. But if there's a flow between those two ponds, now all of a sudden they're going to be more similar because they're mixing. Your textbook also gives the example that, 10,000 years ago, the human population was much more sparse. We had barely any humans 10,000 years ago. We had lots of humans, but they're spread out all over the continents, except for really Antarctica. So the differences between local populations became even more dramatic the longer that they were apart from each other. We have the example in your lab, in your Go Formative, is with skin color. It's fascinating. I love learning about skin color and the evolution of skin color. That's just one of those phenotypes that became very different. Think about face shapes, bony prominences on the face. You can look at the skeletal structure of the face and determine the ethnicity of a person without even seeing any features, just looking at the skeletal structure. How did that happen? Well, it's because for so long we were so isolated and we stayed where we were. Over the last 300 years, though, migration has increased dramatically, especially over the last 100 years. So gene flow, now genes are flowing from one place to another, and altered allele frequencies make us more similar. All right, so gene flow counteracts natural selection and genetic drift. And so natural selection, like we said, that would change the allele frequencies. Members of a population that are better adapted to the environment have greater fitness, survival of fittest. And not only survival, they're more likely to reproduce. Okay, so the ones who are best fit survive and reproduce. Founding concept of natural selection. Over generations, the proportion of favorable alleles is going to increase in the population. So the bird that has the brown feather pattern, or that allows it to camouflage a little bit better against the tree, that one is going to, all of a sudden the population will start to look more and more like that one. Or if we think of, if all of a sudden our lilies, or we put in here some plants that were orange, the ones that were orange would blend in better. All right. So that would be natural selection, but natural selection leads to adaptive evolutionary change. All right. And so it explains why organisms are well adapted to the environment that they live in, but it also helps account for the remarkable diversity of life that we see on the planet. Why one species of bird looks so different from another species of bird, and from another species and another species, we wind up with this mass. The biodiversity is huge because natural selection allows each type of individual species to be perfect for their environment, their niche. Okay. So gene flow and all that, it's going to counteract natural selection a little bit, right? But natural selection is still natural selection. You're still going to have survival of the fittest. So like if that bullfrog had a fourth or a fifth leg and hopped over to the gene pool, yeah, you might wind up with some fifth leg bullfrogs, but if they're not best fit, that trait is going to die off. So that adaptive evolutionary change, natural selection enables populations to change, thereby adapting to different environments and different ways of life. It preserves individuals with favorable phenotypes and their genes and eliminates those, like the fifth-legged bullfrog, with the unfavorable phenotypes. All right. Those who are not best fit, natural selection acts against them. Individuals that survive and produce fertile offspring have a selective advantage. And like I've said, natural selection is not everything. What if the best fit person or best fit organism only has one child or 0 children, but maybe one that's not best fit or it's mediocre has like 20 offspring? Well, the gene pool of the next generation is going to reflect the reproductive success, not just the survival. All right, so natural selection does not act on genotypes. It acts on phenotypes, which are the expression of the genotypes. So natural selection doesn't know what genes you have. It just knows what traits you have on the outside. Phenotypes represent interactions between the environment and all the alleles in an organism's genotype. All right, so we usually think of like an organism's phenotype is all of their traits together. And your phenotype, all of your traits together, is your representation of all the genes that you have. All right, so many plant and animal characteristics are actually controlled by more than one gene. Most traits are not, or characteristics are not Mendelian in nature, meaning one gene, one trait, 2 alleles. Most traits are polygenic. Think human height is the best example of polygenic. All right. And when traits are polygenic, you can get a lot of distribution through, you wind up getting a normal distribution, and it looks like a bell curve. When we graph the frequencies of these traits, we get a bell curve, a nice normal bell curve. But there's going to be 3 types of selection that can cause changes in those normal distributions. And these will probably sound similar or familiar for you guys. All right, so these are the modes of selection. You've got no selection, which would show you this blue part here. This is showing you coloration in beetles. So the blue is the normal distribution, the normal bell curve of individuals by phenotype. This is the original population. When we put this purple part on top, that's showing us the distribution of the population once it has evolved. And the arrows represent the pressure of natural selection on the phenotypes, okay? So in stabilizing selection, you're gonna have the norm, the one that, so this is showing us the number of individuals and the phenotype as a gradient, because polygenic traits are usually like this. So the ones that are in the medium tone color, that's the ones that we have the most frequency of. And stabilizing selection would say, okay, this is going to become even more frequent. And the ones on the outskirts, the really pale or the really dark, are going to reduce and maybe die off. You're still going to get some random variations and some random, you're still going to get some of them, but like it's not going to be 100% fixed here, but you're going to see it become much closer to being fixed. Directional selection is where now all of a sudden this population shifts, and notice that it becomes darker overall compared to the original. Okay. And then disruptive selection, which is much more rare, is going to be where the middle, the one that's the average, actually reduces and the light and the dark become much more popular. It's not terribly rare, but it's relatively rare. Now, for your, let's see here, you have videos. I'm going to post the links here, but let's take a look here.
Imagine a butterfly population that shows continuous variation in wing color. The most common forms are between the extreme forms of the trait. Suppose there is a shift in the environment so that forms at one end of the curve are selected against. Over time, if the environmental pressure remains constant, the most common forms in the population will change. Eventually, forms at one end of the range of variation will become the most common.
All right, so that is directional selection. Let's look. Is there one for stabilizing? So there's directional, then, okay, here's disruptive.
Imagine a butterfly population that shows continuous variation in wing color. Suppose there is a shift in the environment so that intermediate forms are selected against. Over time, if the environmental pressure remains constant, the extreme forms will become more common. Eventually, intermediate phenotypes may disappear from the population entirely.
All right, so that's disruptive. It disrupts the norm. They're named for what they do to the graph. All right, directional shifts it in one direction. Stabilizing stabilizes the norm. Disruptive disrupts the norm. All right, so let's look at the stabilizing.
Imagine a butterfly population that shows continuous variation in wing color. Suppose there is a shift in the environment so that forms at either end of the curve are selected against. Over time, if the environmental pressure remains constant, the more extreme forms will be eliminated. Eventually, intermediate phenotypes will become predominant in the population.
All right, so these are your three types of selection here. All right. And there are examples in your book that I think explain it pretty well. So human, so let's look at stabilizing first. Human birth weight is the most common example I've ever seen for this. So body weight in pounds when you're born, and this is the percentage of the infant population. This is a sample from 13,730 infants, but across the board, this is always what it generally looks like. All right, so the red curve shows mortality rate, and the blue curve shows the normal distribution. Okay, infants with very low birth weight. All right, you're not gonna even have much data around here, but 3 to 4 pounds. Okay, that's the mortality as you get from 3 pounds up in weight is going to decrease. So what we're going to see here is this normal distribution curve, and it looks like the 7 pounds is the norm. And if you think about it, it makes sense. If you're born very small, 3 pounds, 4 pounds, you're usually premature, right? And that means you're going to have more issues developing. Nowadays, with modern science, luckily, it doesn't mean, that the baby's not going to survive at all. It's just the mortality rate is much higher. Now, body weight over this point, okay, now you see the numbers start to go up. 9 pounds, it's higher, but 10 pounds and more, I mean, usually there's something wrong in that aspect too. And you're more likely that baby and mom are going to both have issues. So what this means is that we wind up with a very stabilized norm around 7 pounds. And that's the one with the lowest mortality rate is going to be the norm. All right. So this is a stabilizing selection. It narrows that bell curve. Here's another example of directional selection. So this is when, the environment changes and phenotypes at one extreme become more favored. So it's going to occur if the alleles are favored under new circumstances. So during a drought, this was from this couple. They studied the Galapagos finches in the 1970s and studied them for a long time. And what they found is seasons of drought in this generations following seasons of drought, large seeds were then the primary food source because the smaller seeded foods died off in the drought. So the beaks that were larger, the finches that had the larger beaks, they survived and reproduced more. So we saw the we saw the average trend to the right towards beak size. So beak size being small to large on this graph. And this is just showing you the actual from 1995 when they put all their findings together. You see the average before the drought, weight, average after the drought, the bill length, the bill depth, the bill width got bigger, larger over time. Even the larger birds seem to fare better than the smaller birds. And here's the difference showing. All right, so this is the actual data that from their book, predicting microevolutionary responses to directional selection. Was a published journal article. All right, now directional selection in your textbook also, there's an example of, what do you call them? I'm blanking, bighorn sheep. So bighorn sheep here are hunted. Okay, the rams are hunted and they're typically killed this is in Canada. They're typically killed prior to reaching their full reproductive potential. So they actually contribute less to the gene pool. So they actually look at these huge horns they have. The results of a study was that there was a 25% decline in the average ram weight and horn length over a period of 30 years. So the population, the genes drifted towards being smaller, away from what you would normally think. It's a paradox because the rams that would normally have the greatest fitness because of their size and ability to use their horns and to compete with males were actually less fit because of hunters that singled them out. So this is a directional selection where now it went towards smaller horns being preferred because these guys were just being hunted. All right. And then disruptive selection is usually extreme changes in the environment may favor two or more different phenotypes. Relatively rare, selects against the average phenotype, and it occurs only if alleles favored under the new circumstances are already present in the population. So it's not like you're just going to mutate and create a lighter or a darker version of your average. They have to already have those available in the population, and you get a divergence of distinct groups of individuals, so you can get some like a bimorphism here. And so this is showing you the population of, I think these are another bird before and after this drought, right? The same book, but it's showing you the before and after. And so same thing here, you wind up with this different type of the middle size seeds disappeared after this drought. So you wind up with where the middle ones are out of luck, but the very small and the very large are adapted. And so you wind up with two subgroups. They're the same species still and they can still breed, but they become very different looking from one another, right? Depending on the food source. So, you get this extreme variation or this extreme situation, which causes this variation to pop up. So genetic variation is introduced into a population, we know by mutation, all right? So sexual reproduction does contribute to genetic variation because you're going to recombine mutations in a new way, which may be expressed as new phenotypes. So combining the, sperm and egg or pollen and ovum, whatever it is, you can create something that's never been seen before. And so it's not just about mutation, it's also about sexual reproduction. Okay, so that is one of the main ways we get this variation in a population. And so genetic variation among individuals in a population may or may not produce distinct phenotypes. So when we have something called genetic polymorphism, poly meaning many, morphs meaning like versions of a trait. So biologists may estimate the total amount of what's called genetic polymorphism in a population by comparing different forms of a particular protein. So it's the same protein, but maybe there's different forms, like hemoglobin or something like that. So an example, you can have slight variations in amino acid sequence of a particular enzyme that you can detect with gel electrophoresis. It's the same enzyme, but there's different morphs of it. There's polymorphism within it. So slight variations in the amino acid sequence cause each to migrate in the gel at a different rate. Gel electrophoresis separates by size or charge. So depending on the species studied, they've actually found that the percentage of enzymes that are polymorphic, meaning they're not all exactly the same amino acid sequence, can range from about 15% to about 70%. So if you remember, we said that it's only really if those amino acids are at the active site that makes them, that can be a problem. But the rest of the enzyme can be different, apparently. And this is still very much being studied. But they have found that some can differ as much as 70%. And in general, polymorphism of these proteins tends to be greater in plants than in animals, which is another interesting find. So determining the sequence DNA nucleotides can detect genetic polymorphisms in alleles that differ by only one nucleotide. So your gene for whatever trait versus, all the rest, like if we took our class and we all looked at the same gene on the same loci, the same location, well, how would they all be different? They could all be different by just one single nucleotide. That's called a single nucleotide polymorphism, SNPs for short. They are common in most populations. So most populations, you're going to find one nucleotide being different. Genetic changes in which segments of DNA have been gained or lost, that is called CNVs. Those are copy number variations. So the copy number, meaning a lot of genes have repeating segments. And if those segments are lost or gained, then it's how many CNVs does each organism or individual have. And that can involve DNA segments from 500 to up to 2 million base pairs. All right, so this is still very much fresh research, but currently estimated that the average human may have a dozen or more CNVs in their genome, areas where DNA has been gained or lost. And it does not appear to be associated with a phenotype, or the phenotype is benign, such as maybe an effect on eye color. So maybe little speckles or whatever in eye color. So this there is a link they're finding, this is newer research, of specific copy number variations to increased risk of certain medical conditions such as autism, Alzheimer's, and susceptibility to HIV infection. So CNVs are something that are very much, we're still learning about, but as it's a very important area of research for our medical field. Right, so sometimes polymorphism, you wind up with something called balanced polymorphism. And it's a special type of genetic polymorphism in which two or more alleles persist in a population over many generations as a result of natural selection. And there's going to be two mechanisms that preserve balanced polymorphism. One is heterozygous advantage and frequency dependent selection. So let's take a look at those. So first is heterozygous advantage. Just looking at the words, that means there's some sort of advantage to being a heterozygous individual. All right, so a heterozygous advantage is when natural selection maintains the alleles that are unfavorable in the homozygous state when the heterozygous has a higher degree of fitness than either homozygous. All right, so the heterozygous has the advantage, and because of that, because it has the little a, that means the bad combination, the little a, has to stick around. It winds up continuing in the population even though it shouldn't, because it's a disease. All right, there's a really good book called Survival of the Sickest that's all about this. Let me put that here. Survival of the sickest. All right. So take, for example, um, malaria, or not malaria, sickle cell anemia. Why does a disease like sickle cell anemia still exist if it kills people? For many, many, many years, it was killing people. Now we have treatment, but it can still kill you. Why? Okay, because heterozygous carriers of the sickle cell, if you have the genotypes here, if you have little a, you have sickle cell. If you have big A, you don't. And if you have big A, you're a carrier, you have sickle cell trait. You don't have sickle cell anemia, but you have some of your cells might have a sickle shape. And if you have that, you are more resistant to malaria. So heterozygous carriers of the sickle cell allele are more resistant to malaria, while homozygotes for sickle cell usually die early. All right, so these poor people, unfortunately, this will keep happening because the heterozygous individual wins in malaria. All right, so when malaria is a problem, the heterozygote is more fit than either big A, or little a, little a. All right, so here's a map. This shows us the distribution of malaria. Okay, it's based on elevation. Mosquitoes like certain elevations. So these are where you'd find malaria most. And this is the frequency of the sickle cell anemia allele, red and orange. So you can see that they match up a lot of the places where you see higher frequency of the allele is where we see malaria. All right, so this is heterozygous advantage, and that's going to lead to this balanced polymorphism where we wouldn't expect to find that. All right, and then the last one there is frequency dependent selection, which can also lead to this balanced polymorphism. So a phenotype that has a greater selective value when rare than when common in the population. So think about it like this. It often acts to maintain genetic variation in populations of predator or prey species. So a predator might catch and consume the more common phenotype, and it may ignore the rare phenotype. So this is going to have a different effect on natural selection or on the results of natural selection, because the predator is acting as a selective pressure and it's picking off the more common ones, which would normally persist more in the population, and it's leaving behind the more rare ones because it doesn't know what to make of them and it doesn't want to eat them. So now the less common phenotype produces more offspring, and therefore the next generation has more of those alleles. So they survive more and then reproduce. So all of a sudden now it's actually an adaptive trait. All right, so one example is scale-eating cichlids. Oh, these are so gross. They actually bite the scales off other fish. I have one in my fish tank, I think. But there are two different alleles here. They have a left pointing mouth or the right pointing mouth, and the right pointing mouth is dominant. So we would expect to see it more frequently in our population. But because we wind up with a 1-to-1 ratio of the left versus the right. And the reason is, so this is showing how the cichlid with the right pointing mouth attacks on the left flank versus the other one's the opposite. The approximate 1-to-1 ratio is consistent with frequency dependent. So the selection is dependent upon how frequent that trait is, because prey fish become especially wary to attacks by the cichlid type that is currently most common. Therefore, either type is at a selective disadvantage if it rises above 0.5. If it rises above 0.5 or 50%, the prey start catching on and they're not going to be attacked as easily. So you wind up with this, like, there's more left, there's more right, there's more left, there's more right. And they just constantly balance back and forth because the prey act differently depending upon which one is more common. All right, so it's an interesting case, an interesting example. And we already kind of touched on this, but neutral variation, sometimes variation does not alter the ability of the individual to reproduce or survive. Therefore, it is not adaptive. It's difficult to determine, and an allele that is neutral in one environment may be beneficial or harmful in another. But some variations are just not going to really do much in terms of natural selection. And so then the last little bit here is about sometimes in a population, we actually might have a lot of variation depending on geographic range. So geographic variation, this is a lot in plants and migratory, or I don't want to say migratory, but animals or plants that they take up a large area of land. So they might have a lot of habitats. And so genetic differences among a different population within the same species, such as a cline. So a cline is when you have a gradual change in a species phenotype and genotype through a series of geographically separate populations, and it's as a result of some sort of environmental gradient. So it's common among species with continuous ranges over large geographic areas. That's what I was trying to say. So think about birds. So, hawks and things like that. Body sizes of many widely distributed birds increase gradually as the latitude decreases. I'm sorry, increases. Presumably because larger animals are better able to withstand the colder temperatures of winter. So as the latitude increases, the same species of bird generally becomes larger as latitude increases because have more seasons. A hawk up here, it might be larger than a hawk in Florida, but they might be the same species of hawk. Um, One example of that is the common yarrow. Okay, it's a really pretty little flower. And it grows in habitats all the way from lowlands to mountain highlands. This is the Sierra Nevada range, all right, the Great Basin. And notice that they have a lot of variation because of where they geographically are, but they're all the same species because they can all breed together. But you just wind up with this geographic variation. Now, within one area, like within this area, you might find some tall ones, you might find some medium-sized ones, but in general, the average is shown here. The average is smaller over here, whereas on the lower lands, the average is taller. Okay. All right, guys. So that is it for chapter 19.